Welcome to Stock Therapy with Penny Queen. Today I'll be talking about Medicenna Therapeutics. It's a company that I've been following for several years, and it is the only biotech in my portfolio. I think it is a very compelling case, and there are a ton of catalysts coming over the next couple of months. So I have done my best to make this as light on technical details as possible, but it's a really complicated story. If you like what you're hearing, I have plenty of resources for other investors that have been doing very deep dives. But for now, I'm just going to give my explainer on what Medicenna is, and then I'll lead into the interview with their CEO. So Keytruda is the world's best-selling cancer drug, a $30 billion a year immunotherapy. You can think of it like a missile that helps the immune system see cancer cells. The problem is that it only works for about 30% of patients, and even in the patients who do respond, they often stop responding over time. Also, its patents are starting to run out soon. When that happens, the entire Keytruda franchise needs a way to extend its life and help more patients respond. This is where this roughly $70 million company, Medicenna, comes in. Their drug, MDNA11, is like turning the immune system's volume knob up in a smart way. It boosts only the cancer-killing cells without boosting the suppressor cells that shut the fight down. Older IL-2 drugs blast at everything at once and cause dangerous side effects. MDNA11 is engineered selective, and it stays active in the body much longer. And here's the part that's turned heads. MDNA11 has already shown real tumor shrinkage, including complete disappearance of tumors in patients who had stopped responding to Keytruda and other checkpoint inhibitors. These are end-stage patients whose immune systems were already exhausted from multiple prior therapies, some of the hardest patients to treat. In the monotherapy arm alone, Medicenna saw complete tumor disappearance and multiple partial responses. So the monotherapy is where they are only testing Medicenna's drug. So tumor disappearance, multiple partial responses, plus long remissions that have lasted more than a year after stopping the treatment. This is rare for this class. And now here's a real kicker. A competing IL-2 PD-1 program was just acquired for $11 billion by Takeda, showing how valuable this field has become. But here's what makes Medicenna different. That competing program uses an unproven checkpoint inhibitor and unmasked IL-2 that has never shown single agent activity in humans. Medicenna pairs the world's #1 checkpoint inhibitor, Keytruda, with an IL-2 that's already shrinking tumors on its own in end-stage patients. Big Pharma needs the next add-on therapy that can boost checkpoint drugs like Keytruda and reset the patent clock. Medicenna has 2 shots at that opportunity. MDNA11 already showing single agent response in very sick patients, and MDNA113, their next generation Keytruda booster. So Keytruda tells cancer-killing T cells to wake up, but a lot of them are still half asleep and too weak to fight. MDNA13 is an amplifier that stays quiet in the bloodstream and only switches on right at the tumor site, where it supercharges the exact T cells Keytruda just woke up, while avoiding the systemic toxicity that has plagued other IL-2 approaches. Both are aimed at the same fundamental problems facing Keytruda and other PD-1 drugs. They need to work for more patients, and they need a new partner to carry the value into the next decade. And now Medicenna is taking the next big step. An independent Italian melanoma foundation is sponsoring, as in paying for, a new trial testing MDNA11 before surgery in earlier stage melanoma patients, a remarkable vote of confidence in the science. These patients are healthier and their immune systems are stronger, and this is often where immunotherapies perform best. So if you were just hearing about Medicenna now, here's a simple version. They're building immune amplifiers that the world's biggest cancer drugs may rely on to survive the patent cliff. Early human data is encouraging, and major MDNA results are coming in December at multiple conferences, along with this new earlier stage trial that is launching. This is a moment that people are starting to pay very close attention. Today, I have Dr. Fahar Merchant with Medicenna. Welcome to the show.
Great. Nice to be here again after, I think, 3 years or so?
It's been quite a while, but you have made a ton of progress in that time. So
Yes.
I reached out to some members of my community, and we've been talking about everything that's been going on at Medicenna. and we have a lot of questions. So let
I-
me just launch with, let's talk about MDNA11. How unusual is it for a drug to shrink tumors on its own in patients who already failed Keytruda and other checkpoint inhibitors?
That's really rare because really the patients that we've been treating have failed. In most cases, the patients have had surgery, they've had radiation, they've had chemo, they've had Keytruda, of course, those checkpoint inhibitors, the multi-billion-dollar blockbusters out there. And at that point, the patients, when they enter into our study, these patients do not have any other standard of care options. And in a sense, the option for them is getting some really radical chemotherapy or preparing for hospice, etc. And the response rates for end stage patients who failed Keytruda, etc., is probably in the single digits, 2 to 5%. It's rare. So what we're seeing here with the data that we are sharing and we will share again in December is the impressive response rates that we're seeing in these end-stage patients. In fact, if you look at a drug like Keytruda or Opdivo, both of them, multi-billion-dollar blockbusters, the response rates in newly diagnosed patients are in the sort of 30% range. And when we see that kind of response rates in end-stage patients, it's, really impressive.
Now, you had patients whose tumors completely disappeared on monotherapy. What does that tell you about the strength of MDNA11?
Well, interestingly enough, I think if you go back to history, this is 2, 3 decades ago, when the very first generation of IL-2 was developed and is still available, there was a small percentage of patients, about 5% of patients, that had these sort of long-term cures. So that even after 10, 15 years, those tumors never showed up. It seems like IL-2 has a certain mechanism which is sort of provides that potential for long-term cures. The problem with that IL-2 has been that there's a risk. The drug could potentially kill you. It is so toxic, right? So, not surprisingly, that drug never took off, never became a big drug. And there's been so much interest in coming up with a better version of that same IL-2, which is safe, but gives you this kind of responses. What we've seen with MDNA11 is true in the sense that, to give you an example, we had a patient with pancreatic cancer. This patient had surgery. As you know, pancreatic cancer is incredibly one of the most difficult tumors to treat. The patient had surgery. Unfortunately, the patient also had the tumor metastasized to the liver. And again, liver metastasis is very, very tricky to treat. And the patient then got a combination of 4 different chemotherapies. The patient's tumor continued to grow. The patient then got a cocktail of 2 different chemotherapies. Patient was simply unable to tolerate that drug. It was too toxic. And then the patient got Keytruda, and the patient's tumor continued to progress. And the worst thing is that the tumors were progressing in the liver. So the very first time, within 8 weeks of the patient taking the first dose of MDNA11, the tumor shrank by 25%. By 12 weeks, the tumor had continued to shrink, and within 7, 8 months, the tumor had shrunk by about 70%. Patient obviously felt really happy, decided to go on vacation for 7 weeks and came back. The interesting thing was the liver tumors were gone, but the patient had a new tumor that showed up, and they decided to do a single round of low dose radiation and got the patient back on MDNA11. And the patient got treatment for another 2 to 3 months with MDNA11 and stopped treatment on December 2023. As we speak today, that patient's tumor has still not come back. Although the patient has not received MDNA11, stopped receiving in December, has not received any other therapy, and here we are, nearly 2 years later, and there is no sign of the tumor coming back. So that is impressive. Same thing. Yeah,
That is very impressive. I'm
it is.
sorry to interrupt. That is exciting.
It is. And then the same thing we just saw with another patient. This is a melanoma patient. Got two different lines of checkpoint inhibitors, of course, having failed chemo, etc. Got two different checkpoint inhibitors. The patient's tumor continued to progress. The patient then got MDNA11 on its own, nothing else. And the tumor started to shrink, eventually became a complete response. And that patient did not receive MDNA11 for about 10 months, and there was no sign of the tumor coming back. So again, you have this other patient that's seeing that kind of effect. But surprising thing is that it's not melanoma, but also pancreatic cancer. We've seen the same thing with patient with anal cancer, which is a tough one. In fact, patients typically that receive Keytruda or checkpoint inhibitors, those patients, squamous cell carcinoma of the anal cancer itself has a response rate of about 5%. And true, we just treated one patient, but that patient's tumor just disappeared after 8 weeks. So that was a complete response. So pretty impressive there. So, we are impressed with the data. The potential here for seeing these long-term durable cures could be even more exciting if we took the drug into patients who have a decent, healthy immune system. That's where we can leverage the drug to its utmost full capability.
So that's, my next question was going to be about, those patients who have gone, a year or more. I didn't realize we're almost up to 2 years on that.
Yeah.
So can you explain how important that kind of durability is for IL-2 therapy? I mean, I think it's a bit obvious how important it is, but I'd love to
Yeah.
hear it from you.
Well, the thing is this, that, there are other therapies that will give you a response. For instance, antibody drug conjugates, you'll see tumor shrink, but, it's within weeks or months the tumor comes back. And that is the challenge. What we've seen potentially, if you look at all the analysis we've done on the patient's blood to see how their immune cells have changed, it's remarkable because what we see is the immune quality of cells are different than before the patient entered the study. They are these cancer-fighting immune cells, and we are able to determine that some of those immune cells are memory immune cells, meaning that, even when the cancer has gone, there are ongoing immune cells that memorize or remember the tumor so that if it comes back again, the memory immune cells are ready to attack the tumor. And that's where you get the durability for this particular approach. And other modalities don't do that. IL-2 and particularly MDNA11, the way we've engineered it, it's highly selective for only the cancer-fighting immune cells. So we are able to build a large number of cancer-fighting soldiers, but at the same time, we are able to build these what we call the Navy SEALs of the soldiers, right? So these are sort of your best, most trained, most effective soldiers. And for them to stay in the bloodstream for a prolonged period of time, so if the tumor ever comes back, it's able to knock it down. So it doesn't mean the tumor is not coming back. I think in the pancreatic cancer patient, for instance, the tumor, tries to come back, but it's just that the memory immune cells just knock them down before it sort of becomes large and entrenched in the body. So it prevents that from happening.
Okay. So because MDNA11 works both alone and with Keytruda, where do you think it has the greatest potential impact for patients?
Well, I think in the end, although we are seeing impressive results in these end stage patients, the biggest impact it can have is on patients that are healthier patients. The tumor has patients just been diagnosed with cancer. Why have the patient go through toxic treatments, etc., and destroy their immune system, and then bring in something like MDNA11. So it's best for us to have a healthy immune system. It's like having, you recruit new soldiers, fresh soldiers to fight the battle. You don't go after and trying to see if you can get these exhausted soldiers. It's a much bigger task. It's more challenging. But, that's how we are able to get into recurrent end stage patients. That's how you are able to get the drug into the market quickly because you can get accelerated approval because these patients have no other options. And so while it is important to treat patients who are end-stage patients, the biggest upside is that treat patients early. Immune system is healthy, and this way you can probably delay or prevent recurrence from taking place. And that would be fantastic because then you are potentially edging more towards curative therapy. And even if you can cure a small percentage of patients, you can certainly delay the tumor from coming back by several months or even years. And I think that's where the big impact could be with MDNA11.
All right. Well, this is along the same lines because we need to talk about the Neosite trial. This
Right.
is a trial in earlier stage patients, just what you're talking about.
Yes.
So you're now going to be testing MDNA11 before surgery in earlier stage melanoma patients who are healthier.
Right.
So, and if you could just
Hmm.
reiterate why it's so important, and I guess talking about the timing of
Yeah.
this and the time span of the trial, because I think that's another important thing, because if they're
Correct.
getting this pre-surgery, then the amount of time you're looking at is smaller.
Yeah. So let's describe what this neoadjuvant or the Neosite trial is. This is a trial that's being funded by this foundation in Italy called the Melanoma Foundation. Obviously, they have an Italian name. I won't pronounce it, but nevertheless, it is led by one of the world's top experts in the melanoma field, Paolo Ascieto. He's published more papers on melanoma than anybody that I can think of. He's also chair of our clinical advisory board, and he has been pioneering a number of treatments for melanoma. But the key thing here is what is neoadjuvant treatment? So typically, the first line of defense for every patient that gets a tumor is if it can be surgically removed, remove it right there and then. And that's generally the standard practice. The problem is this, that when you remove the tumor, you don't know if there has been any micrometastasis. Has the tumor spread somewhere else that you can't even see? It might be really tiny tumor spread elsewhere that you can't see on a CT scan or MRI. And this is where patients that have surgery, ultimately the tumor comes back. Although the intent is to cure the patient, we know that surgery does not cure the patient. So now the approach is this. Why don't we have the tumor or the surgery delayed by maybe 6 or 8 weeks and train our immune system to really recognize the tumor so that if there's any micrometastasis elsewhere in the body, you can attack that. And if the tumor ever comes back after surgery, you've already created these memory immune cells. So this way, you are able to delay the recurrence or avoid recurrence for these patients. So that's what neoadjuvant treatment does. So what happens is treatment is delayed by about 6 to 8 weeks. We provide these patients with 6 to 8 weeks of treatment with MDNA11 in combination with a checkpoint. And then 8 weeks later, the tumor is surgically removed. And we are able to see within that time frame. So we get a sample of the tumor when the patient is first receiving treatment. So we get their biopsy. And then 8 weeks later, when the tumor is removed, we get the whole tumor to look at. And it will show us whether the patient's tumor is gone or not. Okay. So what we get is within 8 weeks, we get something called a pathological response. The pathologist at the hospital will determine if 100% of the tumor is gone, or 90% of the tumor is gone, or none of the tumor is gone. And that is really important because that will help the doctor decide, well, the tumor is gone. I can't see it. Should I treat the patient with more drug or should I just stop treatment of this patient? So basically, what it does here is, from our perspective, by using MDNA11, we've already seen this durable effect in end stage patients whose immune system is wrecked. But now, if we are going to treat patients whose immune system is relatively healthy, and if you have the tumor available, then what happens is that you are training the immune cells to recognize the tumor. You have to have that contact between the tumor and the immune system. And it's only then that you generate memory. But if you take the tumor out and then treat the patient with the drug, well, there's no tumor for the immune system to remember or recognize, what am I looking for, right? So that's what it is. Essentially creating, or you're sort of basically taking your immune cells to the garage and resetting your software and putting a GPS system in there and saying, off you go and attack the tumor, right? So essentially that's what you're doing.
So if we're going to use a car analogy, it's the difference between having a tracker on the car before it's stolen or
Right.
having to go out and find it.
Exactly, right. And this way, yeah.
I wasn't even thinking in those terms. I was just thinking about how much more effective it would be and how much less destructive it would be to try and minimize the size of the cancer before. But that's a whole other level of benefit that I wasn't even. Oh,
Yeah. No, and I think that's the important thing. I think that you bring up something very, very important because remember, if now the patient's tumor is 100% gone, do I actually need to do a surgery and leave scar tissue, particularly if a patient has melanoma and the tumor is around their face, on near the eye, for instance? That surgery itself could be complicated. But if we are able to avoid that surgery or even reduce the size of the tumor so that the surgery is now easier to conduct, you are preserving the patient's ability in terms of, living less scar tissue. But think about this. If a patient had anal cancer and have to surgically remove parts of the rectum, just imagine the quality of life of that patient. Or do the same thing with bladder or the colon or the stomach, where patients have to carry a bag of some sort for the rest of their lives. It's horrible. But if we are able to treat the patient with our drug and delay the surgery, or to a point where now you need a minor surgery or no surgery at all, suddenly you've changed the patient's life in terms of better quality of life with less risk of tumor coming back again. That's what the big deal is here, right? So just imagine the healthcare costs and the quality of life improvement for these patients would be amazing.
so what would success look like in this study?
So right now, the standard of care for melanoma, for instance, patients that get this neoadjuvant treatment, they get a combination of a drug called Nevo or Opdivo. This is a Bristol Myers drug. And they get a drug called Yervoy, which is also a Bristol Myers drug. The problem with Yervoy is incredibly toxic. So the thinking is, why don't we replace Yervoy with MDNA11 so that this way you have potentially a better drug, a better combination, and a safer drug. This way you change the treatment paradigm and the standard of care changes for all these patients that are currently getting neoadjuvant treatment but are getting Pembro or other, they're getting Opdivo and Yervoy, and instead they'll get Opdivo and MDNA11. So that's the ultimate objective in this early stage situation. But then you can translate this into other tumors. Why just stop at melanoma? You've got kidney cancer, you've got colon cancer, you've got anal cancer. We've already shown that in end-stage patients, MDNA11 works in end-stage patients with these tumors. If you were to take MDNA11 in a neoadjuvant setting earlier on, then these patients will have that huge benefit of not having to go through surgery, potentially, or have a better quality of life. And finally, the biggest upside is the tumor doesn't come back again, right? So here you are now able to treat, unlike MDNA11 in the recurrent setting, which is you sort of the number of patients is much smaller now. But if you go first line or neoadjuvant, you got a lot more patients. Your market opportunity is massive. And who wouldn't want a treatment that's going to avoid surgery or get a treatment that's going to delay the tumor from coming back, or in some percentage of patients, the tumor never comes back. So that's where the big difference is with respect to what we're doing with MDNA11.
How quickly could results from the study be available since the main readout happens at the time of surgery?
Yeah, so because it's a open label study, we'll get results from the thing is, unlike MRI or CT scans where we follow patients through, it takes several months for us to see tumor shrinkage. Here, we know patients will invariably get the surgery done at the 8-week time point. So within 8 weeks of getting their first dose of MDNA11, we will know that if the patient had a pathological response, complete response, partial response, or no response very, very quickly. And that is important because one other thing is that while the study will be testing Opdivo plus MDNA11, there'll be another arm which will be testing Opdivo plus Yervoy. So we'll have head-to-head comparison between the two drugs. And if MDNA11 and Opdivo is just as good or better than the combo, we think that'll be fantastic because we know MDNA11 is safer than Yervoy. Okay.
Well, okay, so this trial is being run by an independent European melanoma group,
Yes.
and I won't try and say their name either. How meaningful is that for validation?
Well, first, the investigators are top-notch, world-class, who have considerable amount of respect by industry, particularly big pharma. Second of all, it provides additional scientific validation because there you have a philanthropic organization willing to spend millions of dollars of their own money on a project that Medicenna would have paid, millions of dollars on our own. And the other thing is also, unlike Medicenna, to take a product into not first-line therapy. This is not first-line therapy after surgery. This is even before surgery. To take an experimental drug that early, I think we would have had a tough time getting, ethics committee's approval. But with that kind of recognition and reputation that's foundation has and the clinical sites have, that provides another strong validation because they are willing to commit and ask patients to delay their surgery by 8 weeks and try Medicenna's drug, MDNA11, which is not an approved drug, right? So it would take convincing for them to get that kind of approval.
That's really exciting. I have to say that sounds like those patients are the luckiest of the unlucky.
Well, we think so. Yeah, I think that will prove itself as data matures and comes through next year. I think that'll be really good, and I think it'll be also nice to see later on if these tumors ever came back in these patients, right? So that will be very, very strong.
Yeah, I mean, having an armed immune system that knows what to look for, it's got to be an amazing gift, right?
Correct.
So Keytruda is a $30 billion a year drug with patents starting to expire soon. How does MDNA 11 or MDNA 13 help companies like Merck extend the reach of their immunotherapy franchise?
Yeah, so I think there will be potentially biosimilars, copycat drugs of Keytruda, et cetera. And I think it's not only Keytruda expiring, which is a $30 billion drug. Opdivo, Bristol Myers' drug, is also expiring about the same time. So we've got about 2.5 years from now. That's a $10 billion drug. So in all, you've got $40 billion of pharma revenues that are going off patent. And the opportunity here is that MDNA11 could be used in a combination setting in so many different tumor types where Keytruda or Opdivo has already been approved. So in a sense, we think that this particular approach, particularly with the combination approach that we have and the patents we have filed, would provide companies that are developing checkpoint inhibitors an extended patent life. So that's the key thing. And second of all, with MDNA 113, this takes the sort of the program to the next new level where it is really treating patients where Keytruda and Opdivo have no activity. That's currently where those tumors, when we started the conversation, we talked about the fact that these mega blockbusters, they are mega blockbusters. They only help 30% of the patients. The remaining 70% of the patients do not benefit. And this is where 113 has an opportunity to pursue that bigger unserved market, which has a huge unmet need. And I think those two together become sort of a MDNA 11 and 113 become a tag team. They sort of work together in multiple ways, not only for hot tumors where Keytruda is approved, but potentially even cold tumors like pancreatic cancer, brain cancer, colon cancer, liver cancer, where there is really not much out there. Prostate cancer is another big one where checkpoint inhibitors don't work. And this is where the way we've designed this molecule, it's designed to really localize and target those very tumors that are cold tumors, like prostate cancer or pancreatic cancer. And this is where we think there's a big opportunity that is not being addressed right now.
So on MDNA 113, so it's basically designed to stay quiet in the body and only activate inside the tumor. Is that a fair statement?
Well, it's yes and no.
I'm sure it's an
Yeah.
oversimplification.
No, there is yes and no in the sense that the only part that is off is the IL-2 portion. The IL-2 superkine is switched off, but the anti-PD-1 is there. It's on while it's in the bloodstream. As soon as it gets to the tumor, that's where the IL-2 switches on. This is where you need the immune cells to be trained. This is where the tumor has what we call tumor draining lymph nodes. The lymph nodes at the tumor are actually, I call them schools or training center. This is where you go and get educated. This is where the immune cells get to be trained to attack the tumor. So that's where we try and, activate the drug right at the tumor site. And this is very unique because MDNA11 has that capability already built in because it has this albumin component. And albumin tends to accumulate in tumor draining lymph nodes. MDNA 113 gets to the tumor, and by virtue of being in close proximity right at the tumor site, we're able to now train the immune cells to attack the tumor. So that's where, the big differentiator is with respect to how we have designed 113. It is quiet. The IL-2 component is completely quiet, but it switches on once it's at the tumor. And therefore, you don't have to deal with potential safety issues with IL-2 because remember, we are going to be dosing at really high levels, possibly as much as 5 to 10 times higher than what we currently dose MDNA11 with. And therefore necessary for us to keep the drug quiet in the bloodstream and switch on at the tumor site.
Okay, so a competitor working in the IL-2 and PD-1 space
Yeah.
was involved in an $11 billion deal. What sets Medicenna's approach apart?
Right. And that's a really good segue into the potential around the IL-2 space. As you know, there have been so many IL-2s that have failed. The approach that is being used here is very much like a MDNA113 approach. This is a company called Innovent. They just did a $11.6 billion deal with Takeda, where Takeda paid them 1.2 billion upfront, and the rest is milestone payments. Now, they have two components to the molecule, just like we do. We have the anti-PD-1, which is the checkpoint inhibitor, and the IL-2. The thing is that our checkpoint inhibitor is Keytruda, is the blockbuster $30 billion product. We also have a $10 billion version of anti-PD-1, which is Opdivo. So we've got two versions of our IL-2, our 113. One contains the number one checkpoint inhibitor, and the second version contains the number two checkpoint inhibitor in the world. Now, the thing is, when we test both of them head to head in all these mice and so on and so forth, they look very, very similar, because in the end, you want to see something, it might be different in humans. So we are now doing a study in non-human primates, in monkeys, to see which one of these two is better, and then we'll pick one of those into the clinic. So the key thing here is our MDNA113 contains a blockbuster anti-PD-1. Innovent's anti-PD-1 is not the one that's been approved in the Western world. It has not that kind of uptake in the markets in the world. So they're probably working with an anti-PD-1 that's obviously not a blockbuster. Second thing is our IL-2. Our IL-2 has shown single agent activity in end-stage patients and patients who failed a checkpoint inhibitor. Their IL-2 has never been tested in patients. Okay, so there is an untested IL-2 being used in their molecule. Third thing, their IL-2 is not masked, or it's switched on all the time. And the way they've designed their IL-2 makes it look very similar to the toxic IL-2 I was talking to you about. And there is a risk of patients dying with that anti-PD-1 IL-2. Ours, on the other hand, is so much more safer, and we have shown that our IL-2 actually works on its own. So we're taking best of both worlds. We're taking blockbuster checkpoint inhibitors, we are linking it together with a IL-2 that is the only best-in-class IL-2 that has demonstrated activity on its own. So those are the big differentiators. The third thing is we actually localize the drug right at the tumor site where it's needed the most. This is where their anti-PD-1 IL-2 does not do that. So we've got a lot of advantages with respect to localizing and targeting. So you got precision of the drug. Second, we got a PD-1 that's a blockbuster. Third, we got an IL-2 that is showing single agent activity on its own. And none of those features are part of that big transaction that just took place.
So I have to tell you that Medicenna is a very difficult company for me to cover, because like I choose to only keep one biotech in my portfolio. And it wasn't always that way, but Medicenna's data has consistently been impressive. And as the trial sizes have gotten bigger, as there's been more data, it feels like the data's only gotten better. But biotech, as far as stocks go, it's normally a longer story arc. And I knew when I first got into this that, all right, we're a couple years away from really having these results. But, you know, how many coincidences of great results could there possibly be? That's kind of what I've been going on. And then this body of evidence gets bigger and bigger. But when it comes to talking to other investors about it, most people don't have time to be on the phone with me for 4 hours.
Right.
There are a couple that do, and I apologize to them because they've been through it. But trying to break this down, my big question for you is, what's one thing you think investors may still be missing about Medicenna?
Really good question. And I think this is where there has been this skepticism around the IL-2 space because there have been so many failures in the past 5 years. We see that every year. I've literally seen every year one or two IL-2 programs discontinued because they don't see single agent activity. They don't see safety the way they've intended to see safety. And that's where that constant feedback of negative news around the IL-2 is sort of overshadowing what we are doing. And that's where the visibility becomes more and more difficult. You're sort of fighting through the crowds that are against you to try and raise your hands up and say, hey, look at me. We are doing well. And I think this is where we need to do this breakout. And the more clinical data we have, I think there'll be more confidence. As you know, we've got data being released in December at a conference on 10th of December. So that will provide additional evidence of how MDNA11 works on its own and in combination, and what kind of tumor types we're being able to see the drug have an impact on, and what the market opportunity might be for this drug. And I think that's one really important thing. Second is obviously we'll have data throughout next year with the Neoadjuvant study. Scientists who actually, and the pharma companies that want to show me how the drug actually works. Why is it that your drug is working? We'll have biopsies from patients pre and post treatment. That will strengthen the data even more. Third, we'll have non-human primate data in early January from our MDNA113 to show that the drug is a lot safer than the Innovent molecule and potentially much more effective. And I think that will be the third piece of evidence around our platform. And as I say, the second half of next year, we'll be in the clinic with 113. And that should be basically as much proof as we can potentially provide. And if at that point people miss out, something is wrong with me. So I'd say the multiple readouts that are coming up with MDNA11, 113, Neosite, all converge to a point where there will be unequivocal evidence that MDNA11 works. Our IL-2 is the best in class. And there's an opportunity here that could be similar to that of Keytruda and other drugs out there. So that's what I want to try and demonstrate.
So what you just said there is something that I've never said out loud before, because I've always just said, okay, if this drug can make Keytruda work better, that is enough market share in and of itself. That makes it a huge acquisition target. But it's the monotherapy that, when you're doing the ability study, and just for anyone who's not familiar with these FDA studies, you need to prove that your drug is safe, and you need to figure out what kind of dosing is safe. So you start out with a small dose, and when that goes well, you raise the dosing a little more and see the patients there. And the results that were coming out of a safety trial, that right there stopped me. And I went, there was the IL-2 space had been not going well for other competitors. And because there was so much danger in the space, that the stock really pulled back, and you ended up being delisted from NASDAQ. And I'm glad that you didn't waste a bunch of money on trying to stay there.
Yeah.
But it was rough on the stock, and I took that with that ability information coming out, I took that moment and I bought in before. You know, it's all of us in America, a lot of us invest in retirement accounts, and they don't play well with the OTC. So I said, okay, this is my chance, because I'm now seeing monotherapy information come out. And the tiniest potential of monotherapy being possible here is massive.
Correct.
So I couldn't be more. I'm just living in anticipation of your results, because this is proving to be very, very unique, and the safety profile is absolutely amazing. So where do you think you'll be positioned at the end of 2026?
Yeah, so at that point, we've got 4 shots on goal right now. The way we've designed the ability study is we've got 4 different tumor types that look really promising already. So we've got, let's say, patients with cutaneous melanoma who failed checkpoint inhibitors. So we are evaluating that in 2 different groups of melanoma patients. Those that never responded to Keytruda will get MDNA11 and Keytruda together. And those that did initially benefit from Keytruda, but then eventually had progression, they only get MDNA11 on its own. So we'll have those data in melanoma. Then we have patients with a certain biomarker known as MSI-high. So this particular biomarker is found in 30 different tumor types. It's sort of patients with prostate cancer, pancreatic cancer, colon cancer, etc., have that particular biomarker. So the market opportunity there is about 3 times bigger than melanoma. And again, those patients currently receive Keytruda, but then there's patients that will eventually not respond and therefore need some alternative, and they don't have any other alternative. The third group is patients with a tumor type known as TMB-high. It's another biomarker. Again, it's found in so many different tumor types. It's all dependent on the biomarker. That is a market opportunity that's about 10 times bigger than melanoma itself.
Wow.
So those are the 3. The fourth one, we've already seen some encouraging data, is endometrial cancer. And that's something that, again, once patients fail those therapies, checkpoints, etc., they don't have any options. Right now, the only recommendation that the National Cancer Center has for these patients is go and enroll in a clinical trial. That's the recommendation for those patients. Now, that means those patients don't have any treatment options. So if you look at these 4 tumor types, we think that each one of those tumor types has potential, particularly in patients with failed checkpoint inhibitors, to enter or pursue not a phase 3, but a phase 2 registration trial. So you get what we call accelerated approval. You're not enrolling 1,000 patients in a clinical trial. You're enrolling 50 to 100 patients in a clinical trial. So that's the kind of benefit you get, so that you are able to take a drug like MDNA11 and quickly enter the marketplace through the accelerated pathway, where you get drug approved for phase 2, but you could look at entering into phase 3 later on, but you can at least start generating revenue, right? So we got 4 shots on goal, really. Even if you look at a company like Replimune or Iovance, they just got approved. Iovance got approved for their immunotherapy in melanoma, and their market cap is $800 million for patients who failed checkpoint inhibitors. Per patient cost is $550,000 per patient for treatment. $550,000. It's 1 month of hospital stay on top of that. There is 1 in 15 chance that you will die of the treatment because they use Proleukin, right? And it has a market cap of just under $1 billion. So if you now extend that to MSI-high, which is 3 times bigger than the melanoma patients, TMB-high, which is 10 times bigger than a melanoma opportunity. And then you have endometrial cancer, where it's a large population because of obesity. Those are big market opportunities. And if you've got four shots on goal, even getting one of them into accelerated approval is a big deal. Just imagine getting two or three of those, right?
As we're talking right now, the market cap is 68 million US.
Yeah.
Which, as a buyer, I love.
Yeah.
And I love to invest in things that I feel are greatly undervalued. And that's where I see Medicenna being right now. It's, it's the kind of thing where you almost don't want to draw it out for people and show them how big the total pie could be, because
Right.
it's just such large numbers that it's hard to even think about how tiny and overlooked the company is now. But I have a feeling that there's so many catalysts coming this year.
Yeah.
I'm excited. But I want to know, what milestones do you need to hit to feel confident uplisting, and how much runway do you need to avoid financing during a weak window?
Yeah. So at the moment, our runway takes us into Q3 next year. So that's, a not necessarily the healthiest balance sheet, but I think it gets us to all the milestones that we want to accomplish. So the key thing here is we'll have all the data from MDNA11's ability study. We'll have already started, and that's funded program with Neosite. We will be in a position to commence the phase 1/2 trial with the MDNA113. But certainly the company will not be in a position to start phase 2 registration trials with MDNA11 or start the MDNA113 clinical trial. For that, the company will have to raise additional capital. So the key thing here is assuming, and that's a big assumption, that if we don't have a deal done or interested parties that brings in non-dilutive capital into the story, then yes, we'll have to raise additional capital. And the key thing here is, as we've done in the past, because, me and my family are a large shareholder, we obviously want to raise enough capital to get us to the next big milestone and not unnecessarily dilute the shareholders. If we can extend the runway, there are many ways of doing that, but not necessarily over diluting the company to a point where you have got too much cash on hand. And especially if you know that the sort of the big breakthroughs are just around the corner. And that non-dilutive capital is just around the corner. Why go out there and bring in tons of money that you may not need?
I think your holdings is around 22%, you and
Yes.
probably your family.
Yeah.
That means a lot to me. Not just in that you're aligned with shareholders, because if you dilute us, you dilute yourself.
Correct.
And I would love for a group to bring in non-dilutive capital. But I think there's enough that's set to go on that I foresee a lot of movement in the stock, and I can't see other shareholders being upset with a raise that could push this forward. I certainly wouldn't be. And I'd be even more excited if, it ends up being offered to retail. I have a feeling you'd have a lot of institutional suitors here. So
Agreed. Yeah.
yeah.
And I
So
think if you recall, last year we had unsolicited, non-brokered investment from RA Capital. They saw our data at a conference and came to us with an offer that we could not refuse. And again, that's the key thing here, is that if we are able to bring in high quality investors in a similar manner, that's fantastic.
yeah, it was a beautiful raise, and I'm sure they're gonna walk away very happy from their investment here. So I've taken up a lot of your time,
Yeah.
but I really would like to just ask you to talk a little bit about MDNA 55, just to
Okay.
give people an idea, because it I don't believe that the company gets any boost to the market cap for it.
Yeah.
And recurrent glioblastoma
Yes.
is such an important, it's such an important cancer, and there's just really nothing good out there. So
Yeah. So it's been MDNA55 or bizaxofusp that we call it. It's a sort of a mouthful. For those who can't, call it Biza. I think that's good enough. But certainly, I mean, the key thing here is that there has been challenges where data on GBM, whether it's for most of our competitors, including big pharma, those have all failed miserably. And that sort of concern about GBM being a high risk program is continues to be an issue. However, it was interesting when we went to Europe, it's a conference in Vienna just last month, or in fact this month. We had a lot of interest, and there's sort of a revival in pharma companies looking at GBM as a potential area to invest in. So we have reinvigorated our attempt to reapproach a number of potential pharma companies, including mid-sized pharma companies and specialty pharma companies who are looking for niche indications like GBM, and where the opportunity may not be like a mega blockbuster like MDNA11, but yes, there is a billion-dollar market opportunity here with this particular drug. And therefore, we're getting good feedback. There is a wow factor with the data that we share with them, and encouraging to see that there have been two drugs that have been approved, where these drugs are for treating neurological diseases, but the drugs are injected directly in the brain using the same method that we use to treat patients with GBM. So now there is some level of familiarity with drug delivery to the brain itself. And there was previously a concern with big pharma and all of the pharma companies saying, how do you deliver drug to the brain safely? And this is now, showing that there are a couple of products already in the marketplace where the drug is delivered directly to the brain in patients with not life-threatening diseases. These are sort of genetic diseases, but that's where we feel there is a level of comfort now that there wasn't before. When we talk about to companies about MDNA55, and the moment you mentioned local delivery, they would basically be turned off by that. But now it's a different story. They feel that that's the only way, that's the best way, and they're seeing things already in the marketplace. So that's really encouraging.
oh, thank you for taking that detour with me. I know it doesn't get talked about enough because it's not the star of the show right now. But I want people to not forget that it exists, and that MDNA 55 or BIZA could
Yeah,
bring a lot of value to shareholders, as well as making a really big difference for people with recurrent glioblastoma. So,
agreed. I fully agree. Yes.
all right. Well, it's going to be a very thrilling year here. So
Yes.
I will stay tuned, and I'd love to have you back on at some point and to keep talking about this. So
For sure.
thank you for your
Okay.
time.
Thank you. Thank you very much.